Sleep, perchance to replay. Windows into sleeping minds. Özge Yüzgeç. Please.
Well, thanks for being here, guys. Today I'm going to talk about one of my favorite things to do ever, which is to sleep. But I'm going to try to take a bit of a scientific approach to this and try to tell you what we do in the lab and what kind of windows we use to look into sleeping minds. Okay, so as I said, I'm a really great fan of sleep. I sleep anywhere and everywhere, in any condition. Public transport is one of my favorite places, and especially if I'm working on public transport, I sleep even better. It's like a lullaby. But I also don't need a reason to sleep. I can just fall asleep while I'm speaking to you on Skype, which has been captured before a couple of years ago. And I'm actually quite famous for this. I've been known to sleep very well everywhere and anywhere, but enough introduction and things about me. It's not about me, it's about all of us here, because we're actually all quite famous for doing the same thing over and over again. And not only us, it's like all the species out there in the world, whales, birds, your favorite animal, mine is a mouse, and insects, amphibians, all of them. We all spend anywhere from a third to a half of our lives in this thing that looks like coma. We are very vulnerable, we don't move, we don't respond, we don't really do anything. And it seems like we could be doing more interesting things than, like, from an outsider, from an alien perspective, it could look really odd. Imagine, like, when a baby is born, you're waiting for your baby brother in front of the surgery room, and the doctor comes out and says, well, congratulations, you had a really healthy baby boy, but there is something odd about this baby. A third of its life, this baby is gonna be unresponsive, it's gonna be offline, it won't play with you, it won't eat. But worry not, this happens to everyone. But we don't know what it's for. We know that it's harmless. So, we don't know what it's for, it's harmless, but we're vulnerable. To me, it seems like while we can, if you're a whale, go look for your soulmate. If you're a bird, do make your nest even better. As a human, you go party all night. Like, why do you sleep? It seems like there should be something more important than your immediate needs at that moment. Maybe there's something important for a long-term goal. And this is what I'd like to study. But if you'd like to study sleep as a scientist, you need to define it and you need to measure it. So, how do we define and measure sleep? Well, when we're sleeping, all our bodies under the influence of it, let's say. It's very different than wakefulness when you're going about your day. Your heart rate is lower, you breathe slower, you don't move at all. Even there are different chemicals in your blood that is not there when you're awake. There are all these parameters that you could measure, they're all different, but actually the most reliable measure of sleep is up here. It's in your brain. The most dramatic changes in your brain. So how do we measure sleep in the brain? Well, it's a very complex organ. It has about 100 billion cells in humans. They're very densely packed and they're communicating with each other, so we need to first understand what it's made of. A neuron. So this neuron, it has a... never tried to point it. Huh? Yeah, it has a body, and then, as I said, there are billions of neurons and they have to speak with each other, and a neuron receives this communication from other neurons from its dendrites. And then it sends out the information from its axon to the other neurons, the neighboring neurons. And it does it with some electrical activity. And it's so precise that you can, if you have a little voltmeter, if you plug this into the cell, you can measure at what moment and with what amplitude this neuron is speaking to the other neurons. But there are billions of this in our brain. If you want to understand sleep and what these neurons do in the brain, are you going to put millions of voltmeters in each of your cells? This is going to be a big problem. But luckily, in 20th century scientists, they came up with what is called an electroencephalogram, an EEG. instead of going, I'm pretty sure they also went inside the brain, but this is a cleaner way to do it. They came up with this cap that has really that has very sensitive sensors that covers all your skull, and it picks up the electrical signal from basically the dendritic activity of your brain cells. So by looking at this, you can tell in general what's going on in your brain in sleep globally. So what happens to it? They also combine not only the brain activity, but they also look at the muscles when they quantify sleep, when neuroscientists quantify sleep, and they put a little muscle electrode to your chins, and then this is what it looks like. When you're awake, your brain, there's a lot of high frequency activity, it's very fast, things are going on very fast. And also the muscle activity, you're moving a lot. And when you go deeper into sleep, so stage 1, things start slowing down a bit. You don't see these fast things anymore. You start moving less, if any. And when you go deeper, things become even slower. You start seeing these big waves, and there is some burst activity. And when it's slow wave sleep, it's the deepest part of sleep. You see really these big sinusoidal waves, really high amplitude, riding through your brain front to back. And in REM, which is more associated with dreaming, there is really machine-like rhythmic activity, and there's almost complete muscle paralysis. So these are some measurements that we take from sleep. This is how we know somebody is sleeping and at what stage they are. But what is all this good for? What does it do at all? What is its function for you when you go to bed? Well, one of the front-runner ideas in this is that sleep is for consolidation. So we face challenges in our lives. We see big problems, we solve it. We have to learn a new task, you have to learn to play tennis, you figure it out, and you have to remember it. You need to come to this cafe, finding your road through the Lausanne roads, you find it and you need to remember this road for next time. And when we go through these challenges, these steps in our life, we need to create internal representations of the solutions we found, and we need to hang on to them. And this happens actually in our brain in between the crosstalk between these two famous places, hippocampus and cortex. Hippocampus is a deep structure, it's like a learning machine, it helps you learn any kind of task. And cortex is more the computing machine, that covers the outside of your brain. It's more the intelligent, the cognitive part of your brain. And in corporation, they save this information about the solutions you came up with, and they kind of store them in groups of cells in your brain. But hippocampus is too small. It cannot restore all the information, all the knowledge that you have about all your senses experience. It needs to outsource it. And to do that, it needs to, in time, it needs to send it away to distant regions. And finally, these memories become independent of hippocampus and they rest somewhere else. And the slowing down of the brain, as I mentioned just before, these big waves and these high amplitude waves, they help long-range communication in the brain, and this is how you're able to transfer your memories from hippocampus to distant regions of your brain. Okay, this is a very broad way of looking at it, but as a scientist you want to ask, okay, what exactly is the mechanism? How does that consolidation exactly happen? Well, luckily in 2014 some people won a Nobel Prize for solving a part of this question. And it's one of the theories is that the mechanism that we consolidate our memories is through a replay. So you learn something, you repeat it in your sleep. And they tested this by putting mice and rats into mazes. They let them find the way to the cheese or to the chocolate, but they had to try it many times and fail. But once they found the correct path, they went to sleep and repeated that correct path many times in their dreams, which is what they found that happens. And how did that exactly happen? So these scientists in 2014 found a GPS area of our brain, so the place where the location in your brain that tells where you are in the world, and it's also in hippocampus. So if you're going from A to E, Every step you take, I am now in the A spot, and there will be a single cell in your brain that covers only this A spot, and then you move to the next spot, you're a B, but you're heading towards a goal. And each step you take towards this goal is represented by a single cell. And when these cells are active sequentially, take the step A to B to C to E, with this sequence you are guided to the goal that you are trying to reach. And funny enough, when these animals went to sleep, this information going from A to E, because it's the correct way that they have to remember, it replayed in their brain. So these cells, the mouse is sitting, it's lying down on this place, not moving anywhere, but it's still remembering as if it went from A to E. But funny enough, sometimes it was backwards, from E to A, so the place cells were active backwards from E to A. Well, this is not exactly what I do. This is all very inspiring, really groundbreaking science that was done in other laboratories in the UK and Norway. What we study in the lab is a bit different. I'm still interested in learning, but I'm interested in motor learning, in movement learning. How does our brain come about to bring the information from our senses and from our internal drives that I want to move somewhere. I have it's like a mathematical function. I want to get somewhere, I need to move my arm. Should I do it now? Should I do it tomorrow? All this integrates in a function, and then all your senses from outside, your sight and your touch senses, they integrate as a sensory function. And how does this happen? And how does this help us to learn movements? And I wanted to study maybe whether we dream of the movements that we have learned in the day. But I don't do it with humans, we work with mice in the lab, and we teach them to do very precise and very well choreographed movements, and this is what it looks like. So the mice, they sit in a little tube, and they need to start from a very specific location. They need to reach for a target where there's a reward, some sugar water or other liquid reward. They need to follow a really precise trajectory to reach for that. And play this again. They need to follow this really precise trajectory. They need to bring it to their mouths and drink it. And each step of them doing this thing, we measure it very precisely, meaning they can move to different directions and different trajectories. We track how well they move their hands in different locations. And we can even track where their fingertips are, paw tips, their digits are, when they're executing this movement. Because what we are trying to look for, it's no longer where they are in space, where their limbs are in space, and where their body position is at that moment, and we want to compare this to their brain activity. How do we do this? So, one of the windows that we use is what is called a cranial window. So we replace a part of the brain, we replace a part of the skull with a glass window, and we use this very fancy microscope, so we don't use electricity anymore like they did in the 20th century. These are genetically modified mice. When their brain cells are active, they emit light. So by looking with your microscope, at any moment you can see, relating to the movement, what kind of cells are active. So this is what a brain window looks like. And this is what the brain activity looks like when the animal is doing this movement. And interestingly, when they move for different locations, all these cells, they seem to take up a role. So they have a preferred direction. Some of them, they only control the right movement, some only center movement, and some only left movement. And for me, the question was, when you are trying to teach a mouse to do something, their brain looks like chaos when they don't know what they're doing. Their behavior is very chaotic, and their brain is also very chaotic. But in a week's time, they become experts of reaching different directions. And to me, the question was, how does this happen? How does this shift from this chaotic to a very precise and very reliable network that only responds to reaching towards one direction or the other? And can this be happening during sleep? So we started looking at this. And this is what a mouse looks like when it is sleeping under a microscope, depiction. But they didn't quite sleep very well under the microscope. For that, we needed to do some X-ray imaging to find. We needed to do this. What is it called? Like ergonomic study in what position they prefer to sleep, and we found that it's 30 degree. They like to put their head in 30 degree and sleep like that. But another funny thing we saw was that the light of our microscope, it's infrared. You don't see it, but it's so strong that it goes through your skull, through your brain, and comes out of the eyes of the mice. And our camera can pick this up, although the mice itself cannot see this. And what we saw was, when we were trying to look into the sleeping brain through a window glass over its head, we saw that actually its eyes were also depicting another window. In each different sleep state, whether it's an awake or non-REM, meaning this, so REM would be the rapid eye movement sleep, where they're more associated with dreaming. Non-REM is the other state where you would be consolidating your memories. We found that across these states, the pupil size changes really rhythmically. So it looks like, to us, it looked like this is a very precise indication of what brain state the animal was at any given time. And when we quantified its pupil size in red, it's going up and down, up and down. It was the opposite of its sleep depth. So the deeper the sleep, the smaller the pupil. It looked like it was a very reliable, a 90% reliable indicator of what the sleep state was. Well, the interesting is, when you're in deep sleep, you also replay your memories. So it's a very crucial period. You have to, this is when you are consolidating, and you better not be disturbed, because then you might not be consolidating them, and that's a problem. You will not remember the solution the next day. And to us it looked like, okay, then the pupil, is it protecting the brain from this fragile state? Is it protecting the brain from not consolidating? And we wanted to test this with some eye doctor drugs. So this is a tropicamide, is an eye drop that when you drop in your eye, it dilates your pupil. And this is what they use in retinal scans, for example. By flashing light into different eyes, whether it's the small pupil or the dilated pupil, we saw that the animals only wake up if you flash light into their dilated pupils. So it means that when you're in very deep sleep, your pupils are very small, and you're in such deep sleep that if somebody's poking you, if somebody's buzzing the alarm, you will not wake up. And the brain is actually wired to block these information. But somehow pupil is an exception. So when you open your pupils artificially with a drug, then when somebody's sending you light, you end up waking up. But if your pupils are protecting you and you're in deep sleep, you don't wake up anymore. This is, from a neuroscience perspective, quite interesting as well, because it's always theorized that the gating of sleep and the blocking of our senses, it happens really internally. There's a clock in between, a gate in between that blocks different senses, but we also found that it's the pupil itself that blocks this information. So actually, while I was trying to look through a window into the sleeping brain with a glass window over the head, we found that actually pupil is another window that looks into the sleep states and the sleeping brain. And yeah, things in science take a spin like this all the time. You want to find something and you always end up finding something else. But at the moment I'm back to looking through actual glass windows and sleeping brains. And this is what it looks like. So when the mouse is learning something, these are the cells that would be active. So in your window, you would see thousands of cells potentially, but then only a subpopulation are active when learning this task. And we mark them in green. And when they go to sleep immediately afterwards, it looks like it's actually a very similar population that is active here as well. So there's almost a very good match in between the two conditions. So it's no longer how you move in space, it's how you move your body and how your brain represents this movement when you learn it. And then when you go to sleep, the similar information seems to be replaying, just like the people found in hippocampus. So, I mean, just to convince you a bit more that these are, for example, different neurons that you select in your window. And you can find these are just like these are the traces of this. You can take the brightness of that couple of pixels in your image, and you can plot it how the brightness of a neuron changes. And it seems like when they're learning something, they seem to be moving up and down together. If you squint really well, you can actually see it. And when they go to sleep, again, the same neurons that seem to be moving together, that seem to be active together, they seem to be active again together when they're asleep. But we're now, at the moment, trying to find specific rules as to how this takes place. Like, what is the syntax that this replay happens when you learn new movements? Is it a sequence, just like in when you learn in a maze, or is it something else? Currently we don't know, but this is what we're looking for. And so, yeah, what's next for this project? So we looked at the eyes as a window into the sleeping brains, and we wondered, can we do this in humans as well? Can we look at humans' eyes and tell what's going on in their brains as well? And actually, it pretty much works for humans as well. These are my eyes. We were testing things in the lab. So we have these simple EEG electrodes, as I mentioned, that you put on your head and you know how deep you're sleeping. And then we have these virtual reality goggles that cover your eyes so you're in the dark, but they're inside miniature cameras that can track your eye movements. But if you slightly tape your eyes open, you can even track your eyes when you're asleep. And this is the images that you get when you're sleeping. And it looks like non-REM 1, non-REM 2, and non-REM 3, these are different sleep phases, as it looks like, just like in the mouse. As the sleep gets deeper, it looks like the pupil size gets smaller. But this is, to me, the least interesting things to look into the eyes and tell what's going on in the brain. I think what would be more interesting is, of course, at the moment we cannot put a glass window on human brains and look at what's going on in different cells at any given time. However, if we were to do that, it would be very interesting to understand how the rule of learning takes place in your brain, how the thousands of cells in your brain communicate with each other and create these representations of learning, and how can we use this to help people with memory problems, with learning problems. Can we teach their brain, actually, if we know the rule, how neurons should speak with each other and when they should speak with each other, can we actually apply this to people that are not able to learn or that are losing their memories? Or in our case, we study movement and we study movement learning. If we have a prosthetic arm, can we also teach this through teaching things to the brain first, and then you are better integrated with your prosthetic arm, let's say. So with this, I would like to thank you for listening to me and not falling asleep in this time. But if you have any questions, I'll be happy to answer.
Thank you, Özge.
So you said that with the mouses, when the pupil is smaller, it means that the sleep phase is deeper, and when you make it wide, you were able to wake them up by emitting some light.
Yes.
Does it also work with humans?
We haven't tried that yet, because I was not brave enough to... Well, science is a statistical thing. You need to repeatedly do something to know that it's correct. And you go to the eye doctor, get this drop once a year, but if I had to do it on myself for so many times, I had to take it a bit easy. So, well, the question is, it would be very interesting, the answer is, and I think there are some people doing similar things. They're they don't dilate the pupils with drops, but what they do is you again wear a sleep mask, for example, but it has some lights inside. And on your phone there is a movement tracker. If you start moving in your bed as if like you're in light sleep, you're not sleeping so deep, then it starts flashing lights and wakes you up. Because when you're in light sleep, your pupils are larger and you are more prone to wake up. So in a way, it uses the eye mechanism to wake you up, but there's no drugs involved. But I think if you want to try it once or twice, it wouldn't be bad. That's also...
Okay, thank you.
Hi. Right here. Name.
Hello.
Hey. Your name, please.
Mark.
So you agree that if you want to learn something, if you want to learn something, it's better to learn it just before to go to sleep, right?
Yeah, that's what Mom said.
And is it possible to learn something while we are sleeping, or to teach something to someone while he is sleeping?
Yeah, exactly. That would be very, very interesting. I don't think they have done it with humans, but there is actually really cool experiments from mice that they taught them explicitly some things, and it's mostly about location. So I mentioned about these location cells, it tells you where you are. They taught mice to be in favor or be afraid of a location without them having no experience in awake life. So they put them in a corner, and when they're sleeping, they started shocking either the pleasure centers of the brain or the fear centers of the brain. And in sleep, they made the association that I'm in this place, it's bad. I'm in this place, it's good. And when they woke up, they either ran to that place because it was so good, or they ran away from it because it's dangerous. So yes, they have done this with mice, and that would be awesome too. That's would be a great thing to do, because for humans as well, to try to help with memory problems and so on. But then sounds like a bit Inception. It has to be under good hands. It has to be ethical questions are raised as well.
And all of the model regarding the fact that the pupil is open or closed and everything is related to light?
No, all those experiments were in complete darkness.
Okay.
So there's nothing. It just the pupil goes with the depth of sleep. It's the, I don't know, maybe it's like giving the mouse these periods of, okay, close everything, just do things here, don't care about the outside, and then they enlarge, and if maybe a cat is going by, they can see the shadow or something and they wake up, I don't know.
So it would work with blind people or not? Or blind mice?
We haven't tried with blind mice, but I guess it depends on the blindness. If it's retinal blindness, if it's cortical blindness, because this I guess goes through retina, and if their retina is gone, maybe they don't get any light anymore. Yeah.
Hi, my name is Island. Thank you, that was very interesting. I was wondering, because the experiment was very specific to movement, to learning one movement. Do you know if that could be, is it the same as learning from reading a book?
Yeah, I guess these are different psychological categories as well. I mean, by textbook definition, they will be more procedural. Memory, I suppose, the movements, learning a movement. And then this is more like an information. So by category, I think in abstract senses they're quite different, and reading a book doesn't involve so many senses, but when you move, you feel the air, you feel the cold, I don't know, you touch something, you feel it. The factors are different, but I think I'm speculating, probably how you encode it, how you internalize it, and the rules, the graph that I was showing you, I think that would be still pretty similar. It's just the players would be different.
Hi. I had a question. It's gonna come back.
Your name.
Right. So, sorry. Hi. My name is Paul. Very nice talk. Thank you very much. I was curious, from an evolutionary point of view, so we sleep about, I'm very lucky, I sleep 8 hours a night, So that's 1/3 of my life. Sorry. I guess from an evolutionary point of view, this 1/3 is not random. Like, it is some optimization. Do you know whether after 9 or 10 or 11 hours, does my brain stop learning as fruitfully or effectively?
Yeah, it's a good question, these extra hours. I guess it depends on your status at that moment. If you haven't slept in a couple of days, if you have been working for your exams or partying so much.
I don't party.
Yeah.
Just kidding.
Are you partying now?
Yeah, you might need those extra hours, and they could be crucial for your learning. But in your daily case, any regular sleep case, actually it depends. Initial hours are quite important for your muscle recovery, I suppose. And yeah, different phases of sleep that follow each other, they have different roles for consolidating different kinds of memories. Like as your friend was asking, for procedural learning, it's I think it's more earlier in sleep that you recover these kind of memories, and when it's more declarative or like information, that comes a bit after. So each phase is a bit better for another type of memory, but these are with humans, and yeah, you cannot open up and look at cells so easily, so they're a bit more vague experiments. However, these are the hints we have.
Great, thank you very much.
Okay. Yeah, last question. I will come to you.
Sarah. Okay. So I'm playing devil's advocate here. So I'm a scientist, but my boyfriend is not.
So I couldn't hear her.
Okay. So I'm a scientist.
Yes.
My boyfriend isn't. And from a non-scientific point of view, the question is, why are we actually doing this? Because it's super, super interesting, and like, I think we all want to know these questions just for, just why the hell are we spending 8 hours a day in bed? But what's the reason for like making holes in mice's skulls? I'm going to say this is very much the devil's advocate point of view, because I think it's really great and really interesting. But I just kind of want to get your
I mean, I guess as researchers that work with animals, it's one of our duties to speak about these things and why we do these things and where we aim to get to. It's not that it's such a cool scientific sci-fi movie plot and we want to be the actors in it. That's not the reason, obviously. It's although it's at really early stages, these research, it's we want to do it to help with people that have, let's say, memory problems, that have learning problems, and that are losing their memories, and you want to guide them through life by helping their brain remember things with the same techniques that we use for mice. We hope to translate them to humans, so this is our main goal. And I mean, one other thing that we need to really, really emphasize is, especially in Switzerland and many of the other European countries, animal research is really heavily regulated. Every day you need to check their weight, their fur, like their hair, and nobody checks my hair and my weight every day. I mean, they live in really high quality 5-star hotels in the department. Of course, there are always ethical questions to ask, like, is it speciesism? How do we justify this? But this is all, in the end, finally, to come up with therapies and solutions and drugs. Like, come up with a molecule that can activate these pathways that help people learn, or come up with a light therapy that help people restore their memory. So this is the big goal, and especially in Switzerland, we're working very, very carefully on this.
Okay, so I just got a suggestion. So if you or your boyfriend want to volunteer for this kind of experiments, we can discuss this later. Okay, thank you very much, Özge.